So I'm going to invite Steve Rossi to come up and start the next session, which I'm waiving my introduction. You guys can all hear from me tonight. I'll do my talk then. But this session is all about forward-looking. What can we do in terms of driving research? What can we do in terms of clinical trials and studies? And some examples of how we funded research. So,
is this on? There we go. Ooh. Okay. Well, I'll start with just saying we look at this community, and everybody's talked about the power of everybody being together. And the voices that come individually and from you as a group is just so amazing. I've been involved with PSC Partners for 8 years, but really just joined the organization on this side about 6 months ago, and been to many events, and everyone just brings just more power to me in what I'm doing on a day-to-day basis. And one of the things that I'm embracing is how do I take all of this, take your voices, and actually show you what you can do with your individual voice and actually as a group and really make things happen. And as you'll see, a lot of what we're doing with PSC Partners, you've heard through the day how PSC Partners is supporting and collaborating with a lot of research efforts with Sonia and a wide group. But we're doing something also unique as the organization, and it's actually something I've joined for, and it's something that you guys have supported with your voice, with your donations. So much of what we're doing is really important and actually may make some big differences in changing where we go in the future. So there's a concept that seems to be rising everywhere, and sometimes it becomes a buzzword, but I think in the sense of what we're doing, it is important, and it's really called patient-centered research. And it's a changing paradigm, which Gideon referred to, where instead of just the scientists thinking, we have to do this, we have to do that, this is what's important, it's actually going to the patients, what's important to you? And that's where your voice is really powerful. You have to be able to share and express that. And how does it then prioritize what we do? There's so much we can do. And as you've heard, the resources are tough at this point. So identifying what means the most to the patients is key. And there's a variety of ways we can do that. You know, some of it you'll hear is the strategies that we've taken forward with the registry and a variety of things. But the other aspects are bringing the patients together with the physicians, with the researchers. These collaborations are so important in making sure we align and really prioritize what's important, both for the community and even for us as an organization, reflecting and supporting what you guys want. Now, this is not a new concept. It really is, like I said, it's a changing paradigm, and it's going to take some time to really drive forward how the patient even becomes more than just giving input, but actually starts to lead research. And it's interesting that you'll see some of what we're doing at PSC Partners is moving to that late stage instead of just being a collaborator and a supporter and a participant. So an important piece of your voice is participating in clinical trials, participating in research, giving your voice and input to what's a priority. But now we're actually taking that and going into what's called patient-led research, where we actually drive, conduct, and actually do the research with the support of appropriate care providers, investigators. And we've got several projects that we're actually doing right now as PSC Partners that I think many of you may be a part of or will become a part of. But this isn't new, and for many of us who were part of the early days of the HIV epidemic, the patients are what drove therapy to come forward. It was the patients that got in front of the government, that got in front of the providers, sometimes pretty aggressively, but it was needed. And your voice can make a difference. I think we have a way to do that, and this organization is so important in bringing that forward. But it's not just HIV. More recently, we've seen where the patient voice to get care for breast cancer improved. Cystic fibrosis is, I think, where we want to go, where the patient organization has actually driven the development and approval of new drugs for cystic fibrosis with some of the collaborations. And even more recently with long COVID, some of the best publications that really characterized and defined long COVID came from a patient organization that expressed that this is something real, this is what we're experiencing, and it actually helped standardize some of the ways we look at long COVID and where the next stage of research goes. Now, PSC Partners is not a stranger to research, and again, much of your support, your voice, and donations have really helped, I think, move some of what we do forward. Some of the things, including especially in Canada, there's been a participation in our research grants program, which has led to a total of 111 PSC research grants and awarded $6 million of support, and Canada specifically for the past how many years, Mary? Past 8 years have really just contributed a specific grant that Canada funds, which is really key to know that the Canadian resources are actually specifically being dedicated and recognized as from the Canadian Organization for Support. How many of you were at the conference by chance in Las Vegas? Pretty powerful to be back together, and it is such an amazing place for education, just like this, but it's even with broader topics over 2 days, lots of opportunities, and we've been able to sponsor that through the years, and it will continue to be a very important part of our mission. We actually collaborate, and this is where I got to work with PSC Partners, is with the biotech industry, giving contribution to how protocols are conducted. What will a patient want to do in a study? You know, we come up with these great laundry lists of what patients, we got to get this data point and this procedure, and it actually, many times the patients are like, no, this is not, I can't do this. So it's actually been a great piece of input. Helping recruit trials, it's a difficult challenge to get PSC studies actually enrolled. And it's one of the reasons why drugs take so long to get developed in PSC, because the criteria are really tough to get into a trial. And we're trying to improve that, but it's a slow process. So the faster we can get patients recruited and feel comfortable going into studies, the faster we can hopefully see if new drugs can potentially work. We have the patient registry, which I think there's a lot of information, and I'll touch on briefly, which it provides. And then there's a wide range of other places we actually participate, representing the patient voice. Importantly, one of the things we led was the development of an new ICD-10 code for PSC. And that was a big challenge because it was lumped into a bunch of other cholestatic diseases or cholangitis, and you couldn't really understand where were the resources that were being used for PSC, where were the patients, how were they being cared for, and it really allows us to look at that much better in the healthcare system. Now, from some of our initiatives, we went into what's called the roadmap, and I'm going to quickly just highlight this, but the roadmap was really based on 3 different things that came together at once. The first was, you've heard of our Chan Zuckerberg Whereas One grant, and that really allowed us to move into building sort of a research and education program beyond what we were currently doing, and really focusing on how do we bring education like this to the community? How do we bring the stakeholders together? An important one that came after that was the patient-focused drug development seminar. 800 patients from our patient registry contributed to a huge survey that got the voice of the patient in front of the FDA and industry, and really began to characterize what I think all of you know, which is, hey, our lives are impacted by this. Clinical symptoms are a real thing. We really need drugs that are going to work on not only stopping the progression of disease, but also things that can improve our quality of life and our symptoms. And what are the barriers to doing that? And it was a very, very important piece that drove forward where we are doing some of our research now. And a lot of that, as I said, came out of the registry. Now, from that came sort of this whole next stage of really how are we going to bring researchers and patients together to bring education and bring new initiatives forward. And this is really where the roadmap took that forward. And it's still continuing with a lot of our educational initiatives that continue to build upon that. Now, what we wanted to do with all this information is actually, again, do what we talked earlier, is what's important to you. And there's a stepwise process that we really took. It was actually a very methodical way that we looked to take your voices, put it into a really constructive way, and then figure out what are the projects we wanted to take on that would make the most impact for you and would really make a difference. And so we, through focus groups, through the PFDD, we really gathered a lot of information to hear what was a priority and important issue for you. We then took all that, because everybody has a different way of expressing sort of what's important, and tried to categorize those things into bigger buckets of what are the types of research, the areas of research, things that would actually make a difference again, but trying to bring them into places that we could look at big buckets that could kind of capture as many patients as possible and what was kind of characterized for what was important. And we did that through sort of, again, additional focus groups and other ways to bring the voice of the patient into that discussion. And finally, we actually came together with the researchers, looked at the comparison, got their input on it, and it was interesting, as you'll see, where there was some good alignment, there was actually a disalignment in what a good portion of what the patients thought was a priority, and to some extent what the physicians thought as well. And not unexpected. I think you see that on your day-to-day, what you go in with as a priority sometimes, and what gets evaluated or communicated to you isn't always the same thing from a priority perspective. And I think you've heard the importance of communication both ways is very key, and we incorporated that into sort of how we developed our research plan. After we kind of got that sort of what was a priority between the two, we refined those together and really came up with 3 areas that really are not unexpected as priorities. You've heard them today, the things that really make a difference and may move us closer to a cure. The first is really understanding the mechanisms. What's behind the disease, all the things, the complexities that you heard in some of the talks today of how do we better understand those steps that may make the biggest difference in either how the disease moves or how do you stop it from moving forward? What is key in those understandings? The second, and we've heard a lot today again, which mirrors that, is how does IBD and the gut interplay with each other in terms of how the disease progresses? Are there interventions for one that could possibly help the other? A better understanding of that whole relationship, since so many patients have IBD and PSC at the same time. And the last, and this is where we believe is where we hopefully can go faster than what's currently being done. I think some of the work Sophia and Gary are doing to look at repurposing new drugs, but if we can't, or repurposing old drugs, but also helping new drugs to get to a curative phase faster. It's really it can take anywhere from 8 to 10 years to get a drug from discovery to where it might get FDA approved. And there's a lot of barriers along the way. So trying to find faster ways we can do that, safer ways, better ways for collaboration are key. Now, PSC Partners is actually taking on multi-pronged approaches to this. Some of it we're partnering and supporting, and in some cases, we actually took on where something wasn't being done, and we said, we're gonna go ahead and do it. We're gonna find funding for it, and we're gonna actually execute on it. And I think understanding, especially when you give your donations, what actually happens with that, this is how it's playing out. There is so much that can be done and is being done, and hopefully is understood how we wanna make sure what you give, we're giving back. So there's 5 big areas that we've focused on. The first I mentioned are the research grants, and that's really us identifying some of the people in this room have received those grants. It's been a good starting point for launching what the next steps of research could be. But these are seed grants, and these are a good launch pad, but we needed to move from a supportive fashion to actually executing. The PSC Partners Registry, you've heard me mention, there's a lot of opportunities for the way we've pulled it, brought in patient information and tried to bring patient information out to the community, but it's actually a very powerful research tool that I'll show in a minute. The biggest one that we're working on, and is actually hugely supported through the Canadian efforts, and especially Mary, how we got forward and working with community donors, is the WIN-PSC study. This is a unique program that we'll touch base on in more detail, but it's really helping to develop what could be a placebo arm for trials, so that placebo may not be needed in some of the clinical trials going forward. Really better understanding the symptoms. We have a symptom assessment project that I'll touch on. And then most recently, we launched what's called the ICRN, or the International Collaborative Research Network. We brought together thought leaders from all over the world to sit and take what were our research priorities and sit together with patients in the room and themselves and figure out, okay, we've got these areas of research. What are specific projects do we want to take forward? So the registry at this point is, again, a very powerful piece of information, a place to express your voice. I would strongly encourage, if you're not in the registry, it's a really great place to participate if you're looking for ways how you can support the research programs. It's also where the voice is brought together and has a very powerful outward reach. We've got over 2,500 patients in it at this point, and it is a global registry. It is de-identified, so we don't know your personal information connected to you as a person, but there is a fair amount of detailed information that goes over, as you can see, a variety of things from your diagnosis to very specific surveys and responses and data that's cleaned up beautifully by the research team too. So we have a really nice, strong team that supports that and makes sure that the information and works with the community to make sure that information's accurate. There's also a variety of educational information that's on the registry, and encourage you, it's another great place to find out about clinical trials, other patient education, just a variety of stuff to help support you. So especially if you're new, this is a great resource along with the websites for PSC Partners. It's a great place to get some additional information. Now, it's also been a great source of publications. And we are actually starting to get research out there that came from this registry and getting into hepatology journals, being presented at liver disease meetings, getting what we do in the registry out there in front of the providers to really understand the power of the patient voice and how it can actually generate great information that can help drive the field forward. Now, the WIN-PSC study I mentioned, and from a high level, it really was trying to address gaps that we identified 5-6 years ago that just weren't being addressed in either the clinical community, the industry, and even the patient community wasn't sure what to do at this point. And the government and the FDA were clearly not sure what to do. So it actually came together when there was not really something that was moving forward to address some of these key gaps. And one of the biggest gaps is that we don't have an approved therapy. So how can you get an approved therapy? How can you get it through the FDA? The FDA isn't even fully sure about how to actually take drugs through and accept a new drug to come in. But one of the key things that was needed is some of the current ways the drug goes through the system is that you have to do a primary study, and then you have to do a long-term follow-up study. And so after a drug gets approved, they then want people to go on to a long-term outcomes trial that has a placebo in it. We felt that for patients, as much as this is what the FDA wanted, there were some challenges for actually enrolling it and actually some ethical challenges. So we took on as a decision, and this started, I think, when I was volunteering, and Mary championed it, and there were several donors that said, what can I do that's really impactful? And this is what came forward, which is the WIN study. And what we're doing here is actually some key goals. The first is to actually take real-world patient data, so prospective, observational data, all your healthcare information, but collected exactly how it's collected in a clinical trial. So you enroll in this, and all your information is put in, and the goal is that you become part of a placebo arm. So you're getting your day-to-day care just like you do now, but your information becomes a powerful tool so that patients who potentially go into clinical trials can actually get the active drug and then be compared to all your data that you're collecting until you're ready to go into a trial. So it really allows us to hopefully accelerate how drugs may be approved and also, frankly, allow more, I think, speed enrollment up, because patients are more willing to probably go into a study where they know they're going to get active drug. It's just a natural instinct, and I think we all agree that that's probably, if you're going to do a trial, you're hoping you're going to get actually the new medication. But there's a couple of other very important secondary endpoints. Now, we develop these things that are part of drug approval, which are called biomarkers, and those can be blood tests, your imaging tests, potentially even things like clinical things like acute cholangitis or other clinical things that might be an endpoint for a drug or might predict how fast your disease is going to progress. And these biomarkers can help us sort of say that if you have a change in this biomarker, it may predict something down the line, or if it improves, it may mean that you're improving on the drug. We're going to be collecting all this information as part of this study to actually identify where there might be biomarkers that could help us, again, accelerate drug approval. But the last one is a source of data. You've heard a lot about collecting patient symptom data. It actually hasn't been done really well in the PSC setting. There's a lot of diseases that have great symptom, what are called patient-reported outcomes. It's not been done really well in PSC, so we're going to have a large data set where with very specific surveys or tools, we're going to collect information that you report about your symptoms. Very important in terms of helping us support understanding the community and what patients experience in trials, but also it will help develop other tools to better measure those symptoms down the line. So it's a big undertaking. Dr. Hirschfield has been an instrumental part of helping to develop this, but it's a global observation prospective, so it's collecting information not from the past, but going forward. So it's really important. If you have interest in contributing, there is actually going to be a site here in Toronto as well as other parts of Canada, and it will really be an important piece of information that can help us move drug development forward. 2,000 patients. We will be looking at the key endpoints that are sort of associated with the progression of disease. And you'll come in once a year for actually having all your information standardly collected. And then you'll have contact with, the patients will have contact with individual coordinators at the sites quarterly to kind of check in on how things are going. All your lab data, everything would be incorporated into this. We will be collecting all the medications, kind of what we call safety events or clinical symptoms. We'll be doing this very systematically so that it can actually match what's done in a trial. That's what's been a problem with some of the data sets in the past, is that it's a lot of information, but it doesn't match how clinical trial data is collected. And so the FDA kind of looks at it sort of in a, like, it's not quite what we need. So we're trying to address what's been missing out there. A lot of information being collected. I think pretty much all your medical background information, which will be really key to helping us understand sort of what do patients look like when they get diagnosed or come into this, and how do things change over time, and how do those things all relate to each other. So lots of information that can help not us only understand as a community, but especially researchers help understand the disease better. So moving to symptom assessment, how many of you have experienced this? You walk in with all your symptoms and you're feeling like fatigued, itching, and you're just not quite there. It's really just not a good day for you, and you feel like these symptoms are really bothering me. And you start to express it, don't really have a way to express it, or the physicians also just don't know how to quantify it. Not so much the physicians that are here, but especially in the community. And they look at your tests and say, but your tests are great. You must be doing fine. A big challenge we've seen is how do we actually standardize the way we look at symptoms, and how can we measure, can they improve over time, do they worsen, and how do we best collect the information that not only can help us understand in a day-to-day clinical setting, But again, especially in clinical trials, there aren't good tools to standardly measure that. So one of the projects that we've also are collaborating with is led by Dr. Donna Vaughn and Bryce Reeve down in North Carolina. And they are actually trying to take this and actually establish a formal tool that actually can not only better assess symptoms than the current tools out there or improve upon them, but actually then be able to use it potentially in clinical trials so that symptoms might actually be able to become an endpoint for studies. A real important piece that's different than sort of how we develop drugs currently. There are a couple drugs, as Dr. Hirschfeld mentioned, for pruritus, but there's nothing for fatigue. There's nothing for brain fog. And some of it is we don't know how to standardly measure it. So these tools could be really important for new drugs. So some of it is to better understand your experience in living with PSC and the associated symptoms, and do these change over time, and how do we measure that? There aren't a lot of reliable instruments. There's one out there that got developed by a drug company that actually most of the providers and then many of the patients just don't feel measures what they actually experience in symptoms. So it's used, but it's not a really efficient tool to truly understand what's the patient experience. And really to understand how patients feel and function, that is an important piece of what the FDA looks for when we look at drugs. Do they improve how you feel, how you function, but how do we measure it? And so really, if we can't measure those things, it actually becomes a big challenge in actually getting a drug approved. So it may actually help improve some aspects of your disease, but if its main focus is really improving your symptoms and they can't measure it, then the drug won't actually go forward. So it's kind of, again, I won't go through the details here, but there are pretty much five steps, and each step incorporates your voice through very detailed interviews. I'm okay, I'm almost there. It's a five-step process that really starts with, again, the broad interviews. We get more information. You start to develop and refine what you've said and how does it become more standardized. And then every step of the way, the patient voice comes back in. So these are great ways for you to contribute if you want. And there's actually a study that's going on. You don't need to be in North Carolina with the doctors there to do it. There is contact information we can provide, and it's on the website as well. They will do structured interviews. There's criteria for enrollment, but if you want to find a way, again, to contribute about what's going on with your symptoms, this is a fantastic way to contribute to these steps along the way and hopefully have a tool that can actually measure your symptoms and, importantly, might be able to be incorporated into the study that I talked about before, the WIN study, to better measure it within that larger population. This is just sort of the steps along the way. I think I'll skip this one, kind of highlighted with the first one. But I think hopefully this has given you at least a first step of what we're doing as an organization, hopefully understanding where your voice, your support, your donations, and how you can contribute if you want to into understanding how we move the field forward, be a part of research and the importance of research. But also I think it's a very unique opportunity. I've worked in the pharma industry for quite some time. To see a patient organization take on the activities it is, both partnering, driving, and supporting research that can actually help move the field is amazing. And you should be, one, very proud of Mary and the group and PSC Partners, both here, because this is a lead. I wouldn't be standing here if it wasn't for her really, I think, moving this project forward. But there's so much more we can do. And it is a time, you're going to hear a lot about clinical trials next. We're at a crossroads. I've been working in PSC for a long time. We're at a very hopeful crossroads. There's a lot of good stuff going on. You heard with the single cell. All of it's kind of starting to come together at a unique time. So it is important to have hope, and I think we have hope that's much stronger than it's ever been before. So thank you very much.
Thank you, Dr. Rossi, for those incredibly kind words. And we are so lucky to have Steve leading the execution of the PSC Wind project with his decades of experience. And our advisors who helped us design this project, one of whom is Dr. Hirschfield, who I welcome back to speak about clinical trials and studies.
I'm just reading the instructions, Meg. Okay. I'm not touching anything. I didn't bring a joke. Very good. Okay, thank you very much. So for this talk, I've been asked to really focus more on clinical trials and studies. And so what I'm going to do is just, again, give you my concepts and high level about where we're at. And I want to sort of give you some examples from some of the trials that have been conducted for PSC, just to show you what we're doing and to just comment on some of the challenges, but some of the opportunities and some of the excitement. So again, the disclosures. Our patients don't have a lack of need to take part in clinical trials, and it's not difficult when I review what happens to our patients to think about what I'd like to prevent. I'd like to prevent the patient needing an ERCP. I'd like to help make sure their colitis is well managed. I don't want them to bleed from varices. Just see, so I'm going down the side here. So there's your ERCP, there's some of your colitis, there's your varices. I don't want you to have a spyglass cholangioscopy looking for, bile duct cancer, and I don't want a shadow to arise on your MRI scan for us to sort out, or for there to be complex bleeding that we need to fix. So we know what we want to try and prevent, but developing drugs for this is harder because these things take a long time, and we need to find ways to measure the impact of drugs long before this has ever happened to you. And so it's clear why research is important, and the treatments will only come with understanding what causes disease, and that's what you've heard about today. And that is going to be a weak link, and that's what we have to work on, okay? And we know the questions, and we know that your participation is absolutely essential. Why me? Why children? Why men more than women? Why the symptoms? We've heard about symptoms a lot today. Why the bile ducts themselves? We're not even really sure. Why is it linked to colitis? Why cancer? Why does the disease come back after transplant? And then why so difficult to treat? And then importantly for this part of the talk that I'm giving today is, who should we put into the clinical trials? We can't put everyone into clinical trials. We've got to put what we would use is a technical term, informative patients into clinical trials. Because if I put somebody who's about to have a live donor transplant from Dr. Salzner in a month, then no drug is going to work, and that's not informative. Whereas if I flip it and I put someone who's got such early disease that nothing's going to happen from a sort of endpoint perspective, I'm not talking about symptoms, but from an endpoint perspective, for 30 years, they're also not particularly informative. And we need to use our money wisely so that we make success begets more success. And that's why it's actually quite hard to do studies, and sometimes patients feel frustration because not everybody's eligible, and we have strict inclusion and strict exclusion criteria, but they're there for a reason. There's been lots of prior efforts. We've been at this for a while, and people have tried all of the drugs that are obvious to them to see if they'll help. This was a table we did for a review nearly 10 years ago now. We've tried immunosuppression. We've tried steroids. We've tried the drugs that you get for your colitis. We've already touched upon ursodeoxycholic acid, okay, and we can touch a little bit about antibiotics. So what's the common theme is that apart from the ursodeoxycholic acid trials, there are very, very small numbers of patients included in those studies, which means that it's quite hard to get definitive answers one way or the other. That's why we hope by working together we'll get better at it and our studies will improve, and maybe, as I was talking to someone earlier on, we'll do fewer studies but fewer better studies and more likely to have success. And URSO, the most well-studied of drugs, there's about, 8, 9 studies, but even still, the number of patients included is actually under 500 patients with PSC who've ever been in a clinical trial of ursodeoxycholic acid. And you can see that's a challenge. But more importantly, the story of URSO actually should encourage you to take part in clinical trials. Because when we do a study, we are interested in efficacy and we're interested in safety. And in fact, safety comes before efficacy. Everybody told us that we should use ursodeoxycholic acid in PSC because it was obvious. We knew that it works. Be an early adopter. You know the answer already. You don't need to do the study. It works for another disease and improves the liver tests. However, Keith Lindor, was a retired professor at the Mayo, was interested in the dose of ursodeoxycholic acid and how that would impact outcomes for patients with PSC, because one of the hypotheses was we weren't giving enough. And surely if we gave more, it'd be even better. So he did a very hard study across the US of what we call high-dose urso, 28 to 30 milligrams per kilogram, versus placebo. Someone needs to be the placebo. We need to thank the patients in the placebo arm, because in this clinical trial, the high-dose urso made PSC patients sicker, not better. Okay? Now, that's science. That's, it's again, not necessarily on an individual level. You only pick this up when you do the study and you actually analyze it. But the message was placebo was essential to prove that high-dose urso is not a good idea in PSC. And in fact, for somewhat complicated reasons, it made the disease worse. So that's how we learn in medicine to then impact what we do in clinic. So we are all committed to trying to use evidence-based medicine. We look at what's going on in the laboratory, okay? So we look to our colleagues in the labs at the Mars building and around the world, okay? Certain kinds of mice, if you make a mice not have a bile acid transporter, they get a disease that looks like PSC. It isn't PSC, but it's a disease that looks like it. That helps us say, well, then we should think about drugs that affect bile acid transporters and make bile less toxic. We look at other science. So this is a study not going on in Canada at the moment. It's a study going on in the US and Israel and in Europe, where they're blocking what's known as a chemokine. That's a marker, that's a molecule that gets released when cells are sick. But what happens when cells are sick is that actually, particularly when they stay sick, is they release chemicals that say, hit me harder. Okay? And when you hit the cells harder, you do more damage. So actually there's now a clinical trial blocking this particular molecule to see whether that would actually help reduce the injury in PSC. So we are using science to choose the clinical trials. We're not only going to develop treatments for PSC from repurposing. I think that is important, but we'll also need some innovation and some new things. To speak to the point about who should go into clinical trials, that's become something we've become better at. We've learned that we need to be a little bit choosy as to who goes into clinical trials to try and make those studies as informative as possible. If we reverse the journey, we kind of know that if we're going to do an itch study, which is very important, there's no point putting someone who's not itchy into the study. That is not going to help work out whether the drug works. So you wouldn't expect us to put a non-itchy patient with PSC into a study looking at itch. The same now goes with the concept of, in your journey, when are you in the so-called latent stage? When are you in the rapidly progressive stage? And when are you in the stage where you're going to need a new liver? Okay? And we probably want to catch you in the sweet spot when you're in the progressive stage, because that suggests that's a time when we could measure that we slowed it down, we put the brakes on. And some of the work we've done in Toronto, and we've also validated with the Mayo, and we're going to publish it. I hope it's in my long list of papers to publish. It's just identifying that cohort of patients who are on I think about it as the slippery slope. Once your bilirubin is elevated, you're more likely to have an event in the next 10 years. If your alkaline phosphatase stays elevated compared to those patients where the alkaline phosphatase is low, that's a group of patients who are going to move forward quicker. That's important because when we then talk to our sponsors who sponsor these clinical trials, we help them work out who to include and who to exclude. We'll tell them, don't put the patients with the bilirubin too high, don't put the patients with the alkaline phosphatase too low, because they're not informative. We then try and work out how it works without waiting for you to have the event, without waiting for you to need a new liver or have a varus or bleed, etc. And we try our best through consensus to work out what is the best so-called surrogate endpoint. The surrogate endpoint is a test that you do that predicts what's going to happen to you in 10 or 20 years' time. They're not that easy to develop. They're easy to conceive, but they're not that easy to develop, because you can imagine, between now and what's going to happen to me in 20 years, I hope there's a lot of things that I don't know that are going to happen, and therefore measuring something today and telling me that in 2043 something's going to happen to me is not that easy. But as a group, we've talked about the roles of blood tests, the roles of liver biopsies, and the roles of things like FibroScan and MRIs. Whoops, let's test your technology and see if we can go backwards. We can go backwards, okay? In terms of the drugs that are currently being thought about for PSC, we have remained very traditional and conservative. We are looking to persuade people to come in with really innovative ideas. And I'm not going to talk about the super innovative ideas, but there are companies out there who are thinking about cure, who are thinking about nanoparticles, who are thinking about cell therapy. Really difficult studies to do, but they're thinking about it. Traditionally, at what we've been at the moment is looking at drugs that improve bile acid flow along different pathways that we've used in other diseases, drugs that change the microbiome, and drugs that are anti-fibrotic. So I'm just going to give you a flavor for some of these. This is the kind of clinical study where it's retrospective. We asked, it wasn't me, it was colleagues, Cynthia Levy led the study from Florida, and a physician who's now in Australia, Kate Lynch was the first author. She asked everyone around the world, do you have patients who've been on vedolizumab, Entyvio, with PSC IBD, and did it make a difference to their blood tests? So this is what's known as retrospective research. It has limitations because there's a lot of recall bias. People only remember the good things, okay? And what I like to use this slide to show you is that we're optimistic people, but we're honest at the same time, okay? So the simple answer was vedolizumab doesn't help patients' blood tests in PSC. But, we looked at it, there were 20% of the patients, the ALK phos went up, 40% of the patients, the ALK phos stayed the same, and 20% the ALK phos went down. And to reassure you that your clinician colleagues are optimistic, what they wrote in the abstract was the bit at the bottom, that the 1 in 5 patients who appeared to get benefit. So we are optimistic, even though realistically this was not a winner. When we talk about it, we try and at least message it carefully. This is an interesting study which results will no doubt appear soon over the next 6 months. And this is an oral inhibitor of fibrosis. It actually works very similar to vedolizumab, which is what's known as an integrin inhibitor. So, when your cells talk to each other, it's lock and key. There are molecules on 2 cells, and they basically sit together snugly. On the biliary epithelium, you express some of these so-called integrins, and they are actually important when they interact at generating molecules that are then pro-fibrotic. A pro-fibrotic molecule could be something like what we call TGF-beta. It's a molecule that is pro-fibrotic. So this is a clinical trial that some of you potentially have taken part in because it's global, and we're one of the sites in Toronto. And it's been looking at an oral version of a drug that blocks this. And the results will be announced, I suspect, soon, and we'll have to wait for the full analysis. Why are these studies hard? Because when you start a study like this, you don't actually know what dose to work with. So part of a study is actually to work out the dose of the drug. We don't want to take away drugs that might help patients if we haven't even dosed the patient correctly. We have an idea of what dose to give from mouse work, from primate work, from phase 1 human trials. But then when you try in every disease, you've actually got to work it out again for that disease. There's no point presuming that your drug is not effective if the drug didn't even get to the liver. It's got to get there to have its effect. So one of the challenges and one of the things we rely on our patients is that many times when you take part in a clinical trial, it is pure altruism. You are not likely always to benefit yourself from that early phase study, but you're going to provide an incremental knowledge. At some point, you may get into a study that's later phase, and then you might see the personal benefit. Norso is a drug that's interesting because it's being developed in Europe only. It may one day come to the US and Canada. There's a phase 3 clinical trial, and this is like ursodeoxycholic acid, but it's been chemically modified. And when it's chemically modified, what it does is essentially push out more bicarbonate over your bile ducts, so it essentially soothes the bile ducts to make them less sore. And in a phase 2 clinical trial, there was a change in alkaline phosphatase. And there's now a big phase 3 trial in Europe, which is fully recruited, and it did require our patients to agree to do liver biopsies, okay? Because the endpoint was not a blood test, but the endpoint will be, did your liver get better under the microscope? Sounds easy. First of all, you have to persuade patients. Understandably, it's not particularly popular. Second point is, it turns out looking at liver biopsies down the microscope is really, really difficult, and the pathologists can't agree. Even when they're all very good people and you put them in the same room, they'll give you 6 different answers as to how much scarring they see. So not as easy as it sounds. This is a study where I first met Steve Rossi, and he reminded me at least 10 years ago. And this was a phase 2 study of a really interesting drug that essentially has been chemically designed in the laboratory to improve bile flow. And the reason I want to show you this trial just in a little bit of detail, not a huge amount of detail, just a little bit of detail, was it's a 12-week study, 2 doses, placebo, 62 patients from 27 sites. So Steve traveled the world, okay? And 27 sites, and we got 62 patients. It's really hard, okay? And during that study, we have ongoing dialogue discussing the inclusion criteria, the exclusion criteria. Can we do something to make it better for patients? How do we make sure we can actually finish this on time so that we can keep momentum? Now, the reason the study was interesting is that when we talked about the study before it, we expected the blood test to get better, but alkaline phosphatase didn't get better, but other tests did get better. We saw improvements in inflammation, okay? And more interestingly, we saw some improvements in markers of fibrosis, markers of liver damage. And this made everyone then think about how do we design the next study and what do we do in it. Okay, we've tried drugs for PBC, which were like the sister disease, and they didn't really help. But I mean and then other companies made a different version of it and tried it again in PSC. Okay, and there was some early data that suggested it helps. And again, that data was interesting, but it wasn't perfect. That's why you then have to get our patients to take part in phase 3 trials. So this is the results that was presented, it's not been published yet, in April of this year, of a phase 3 trial that some of you could have taken part in, Gilead, where hundreds of patients around the world tried this drug that improves bile flow. When they do that study, again, they ask some of you to be on placebo, they ask you to have liver biopsies, and they design it with what's known as an interim analysis so that they can find out early on whether the drug is working. But more importantly, if it's not working, the patients want to move on to the next trial and have other opportunities. Unfortunately, when you do the bigger study, this trial didn't work. So the first primary aim of the study, the fibrosis didn't get better. That's not to say that the knowledge that we'll gain from that study is not helpful, because we will, okay, when the data comes out, because we'll get better for every study afterwards, but it's to show you how hard it is. I should talk about vancomycin briefly. That is an important drug to trial. It is something that's quite sensitive in the PSC community. We don't know whether it helps or doesn't help. It certainly does seem to help colitis, whether it helps the liver. And the only way we'll answer that is through clinical trials. So, there's lots coming, and I want people to remain very engaged in clinical trials. I want clinicians to always ask, why is the patient not in a study? But I also want you to recognize that it's really difficult. This was a study that we did ourselves in the UK of a really novel molecule, and it was incredibly hard to recruit 19 patients, to give them a drug which they didn't, really didn't have much information on it. But even that small increment does help. And this is what's going on at the moment for clinical trials in PSC. A number of studies for itch, a number of studies for disease modification. So as to keep us time and not get into trouble. Okay, I'll stop there.
Thank you, Dr. Hirschfield. There's a lot to talk about. And if any of you are interested in getting notices directly to your inbox about potential studies that you might be eligible for, please be sure to check out the PSC Partners Patient Registry. And we list on that site both publications related to ongoing clinical trials and also all the clinical trials in North America, at least, that are actively recruiting. So PSC Partners Registry.org. And next, I would like to invite Dr. Zhao, and I don't know if Dr. Bott is here. Yes, come on up, please. Dr. Zhao received his MD from McGill University, is trained in internal medicine, gastroenterology, and transplant hepatology. He's currently enrolled in a program in clinical epidemiology at U of T under the supervision of Dr. Bott. And he is going to speak on a study that Dr. Bott and he are working on that is partially funded by a PSC Partners Canada research grant, so funded by all of you in the room. So.
Hi, everyone. Pleasure to be invited to this talk. And I'd like to thank PSC Partners for funding our research, as well as Mary for being along every step of the way to look at our paper to make sure it's in line with the goals of PSC Partners. So I'm going to talk about the study that we did, looking at personalized evaluation for PSC in patients on the transplant list using machine learning. So as you all know, liver transplant is considered the only effective treatment for PSC. And as we illustrated today through the talks, the current allocation system based on the MELD score is not, it was not designed for PSC, and consequently does not reflect disease severity and does not consider any disease-specific complications, so it performs quite poorly when we're looking at certain diseases, mainly PSC. And patients with the disease depend on living donations for liver transplantation. This is a publication by Dr. Bott recently this year. We're looking at our data. And I want to bring your attention to this sort of unfortunate marriage of circumstances where we're doing excellent with outcomes for living donation. However, patients who do not benefit from a living donation end up having a higher risk of mortality, and as they're not prioritized for the deceased donor waitlist. So what is machine learning? Briefly, it's when you program an algorithm to make decisions based on data without explicitly telling it what to do. So you let the machine figure out what you're looking for. So this is interesting, and it gives you the ability to find nonlinear relationships because you don't have to tell the machine what exactly to look out for, and it will bring to your attention things that you have not considered previously. And it's able to generate more accurate predictions than conventional biostatistics. Recent publications also showed that we have the capability for a comprehensive tool in risk stratification in transplant recipients. So we thought we should apply this sort of technology to the wait list for patients with PSC. So the hypothesis of the study is that machine learning algorithm can make more accurate predictions of waitlist outcome than the MELD score. And we will build such a machine learning model, including PSC-specific variables to predict outcomes. We defined the outcomes as all-cause mortality, transplanted. They were censored at the time of transplant, or patients that we just called censored based on the fact that they were removed due to clinical improvement condition that are no longer required liver transplantation at that time. Initially started with the SRTR, which is the Scientific Registry of Transplant Recipients in the United States. Identified 4,666 patients with PSC from February 2002 to December 2020, corresponding to the era when the MELD score was adopted. And we validated it as well in 144 patients from the UHN. It's important to note that this study was specifically interested in outcomes related to PSC as a disease. So anything that would compete in terms of mortality or outcomes, such as a cancer, MELD exception points, or hepatocellular carcinoma, they were excluded. I gather this is a busy slide, but briefly, we built 3 models. First, with the machine learning Cox proportional hazard. It's a function that's that's pre-built, just sort of as a baseline, and then a random survival forest. Essentially, how it works rudimentarily is generates decision trees, and you can have as many layers of these decision trees as you program it to, and ultimately it's both based on the voting system to generate the result. And the deep serve model, which kind of is it's neural network, so it mimics the function of the human brain. Every point you see on this, on the little dots on the left side, on your right side, yeah, can be seen as an analogous to a neuron. It gets inputs from data, and it's based on the integration of this data, decides whether to fire or not. And then the integration of all of this network, as many layers of nodes as you want, gives you a decision. And we were looking at concordance index. So grossly concordance index is how often does your prediction match the observed outcome? So essentially how precise your model is. So we did this in several steps. First of all, we tested all 3 models on, we trained and tested on the SRTR. The data splitting is a standard thing that's done in machine learning, 70%, 30%, and 10%. So you trained on 70% of the data and then validate and then test on 10%. And we did a cross-validation, so we make sure that not every model is trained on the same, constantly trained on the same set of 10% of data, just to make it more less biased, I guess. And then we took the two models from step 1 and then trained them on individual OPTN regions. So the SRTR is divided in individual regions of donation in the United States. We're thinking, how can we make our model more accurate? So let's see what the SRTR does if we divide them into individual regions. And then we took the best performing model out of those 2, and that's when we introduced UHN variables. So the data set from the UHN, 144 patients who had variables that share with the SRTR. And finally, we introduced PSC-specific variables only found in the SRTR. There's a list of PSC-specific variables, but they include complications of PSC, cholangitis, concomitant inflammatory bowel disease, dysplasia on colonoscopy results, and so on. And finally, we analyzed the SHAP values, which is also another validated methodology in machine learning where after the model is built, you break down individual components of the model to see which variable contributed the most to your model. I'm going to go through all of that. So comparing the SRTR and UHN, you can see that it's a male predominant disease. Our MELD score was a little bit higher than the SRTR, but otherwise comparable in terms of age, BMI. So out of the 4,000 patients in the SRTR, death was around 20%, 11% for the UHN. And the mean time of transplant, 284 days, 250 days. Overall, there were 63,000 follow-up visits in the SRTR and 4,400 follow-ups in the UHN. So we had quite a lot of data to work with, mostly due to the SRTR. And the mean follow-up was 449 days and 348 days, respectively. So this is the step 1 when we compared our 3 models. So the Cox proteome hazard was not supposed to be white on white, but it's there. But as you can see, this is, again, concordance index is how accurate is the prediction that you're making to the actual event. And you can see that all 3 models, almost all 3 models, there's some overlap, but outperform MELD sodium and MELD 3.0 for PSC on the SRTR. So we took the 2 best performing one, which is the Cox proteome hazard and the random survival forest, and we ran it through the individual regions to see if there's a better performance when we break down the data further. It's a busy slide. Essentially, you just test every region with every region and then on the database. But that tells us essentially that there are 2 regions that performs better when we train on them, region 4 and region 8. So I think at this point we squeezed as much as we could have from the SRTR database, so it's time to introduce our own data set from the UHN database. And when we do that, you can see the performance improves even better. We'll call this a fine-tuned model with added UHN variable. I know it's not the most elegant name, but it's descriptive. And you can see that now the performance is dramatically better. MELD sodium hovers around, it kind of performs pretty well in the first 30 days, 0.7 concordance index, but it never really really goes beyond that. Whereas the fine-tuned model, we're reaching about a concordance index of 80%, 0.8, yeah. And now if we introduce PSC-specific variables to our data set, which is kind of the most important part here, the performance dramatically improved, right? We're hovering around 0.9 at this point. We have to be mindful about this because we're only training on the 144 patients because the SRTR and the UHN does not share all the features. So in this step, this is validated and trained on the 144 patients in the UHN. And finally, let's see what's contributing most to the model. So this is the Shapley values. The way to interpret this is red is a higher score of whatever is described on the left-hand side, and then we can see that the MELD score is the most important contributor to this model, which is kind of a relief because if it's not the MELD score, then what have we been using for the last 20 years? But we can see that the addition of other variables contributes significantly to improving this model. Is that my last slide? Or is it frozen? Oh. So this is the example of a patient that we pulled from the from the UHN, just to illustrate how this algorithm works. We had this patient, 57-year-old, who had a MELD score of 15. With a MELD score of 15, it's hard to prioritize for a deceased donor living transplant. So if this patient was seen in the clinic, the probability of being transplanted with MELD score of 15 in Ontario, unless you get exception points, is quite slim. So the MELD score is not quite informative in this context, but if we apply the machine learning algorithm, it can give you a survival probability that decreases significantly. And unfortunately, this patient passed away at around the mark where the survival probability was hovering around 0.6. So in conclusion, I think this is an important proof of concept study. So we know that our model outperformed MELD. And I think the key message is that MELD alone is no longer sufficient. We can do better. We have the technology to do better. And therefore, we should look into this more. It emphasizes the importance also of using disease-specific factors to improve predictive models and not just limiting yourself to the 5, 4, or 5, depending on which version of the MELD you're using. And the future study should look at disease-specific data for each etiology and try to harmonize this for all etiology of liver disease. So we can have actually a better stratification of who is at actually higher risk for poor outcomes on the wait list, so we can prioritize them better, which is what we're working on in the DynaMELD study. So thank you for PSC Partners. Yeah.
Thank you very much. We have about 10 minutes for questions for the three speakers, and Jay Vias, who is father of a young adult living with PSC, is going to moderate the Q&A.
The speaker's going to be right up there. Okay, great, thanks. All right, thanks. Let's get started. Thank you for those informative and insightful presentations. Let's see the questions for all of today's speakers. So maybe it's even more than the four of you. Why did you decide to focus on liver disease and specifically PSC? That's the top question right now. Yeah.
We care what you care about.
Yeah.
Yeah. Is this on? Then you can't hear me. My story is very easy, actually, because I was a medical student in the University of Cambridge in the UK. And when you're a medical student in the UK, you start off by learning medicine. So the first team I worked with was the liver team, okay, and they were somewhat good. And then the next team I worked with was Sir Roy Kahn, who basically was the founding father of liver transplantation in Europe. And so then it's pretty easy where you go from there.
I guess, well, the focus of my research hasn't been on PSC, admittedly, but I'm very good friends and colleagues with Gideon, and my research program, I'm very fortunate to have wonderful trainees like Jun on my team, but we have been dedicated to using novel tools, so artificial intelligence tools in my program, to integrate clinical, laboratory, and omics data to inform personalized predictions. And one of the key projects that my lab has been driving in the last few years is to look at revamping the waitlist prioritization system. And certainly, we both felt strongly about looking at the PSC patient population as certainly a disadvantaged patient population on the wait list. So as you may be aware, the MELT sodium relatively underweights the bilirubin, and by a factor of 3 compared to the INR and the creatinine. So those are parameters that are in that MELT sodium. And we know that the confluence of certain factors, like say female PSC particularly, may be disadvantaged in terms of access to deceased donor organ offers on the wait list. And so we certainly felt strongly that we should look into this problem and try to make this a more equitable prioritization system, not only for PSC patients, but also PBC and females, whoever is disadvantaged on that wait list. And I think the interesting thing about AI tools is that we have this ability to really understand the dynamics of data on the wait list. And certainly we know that PSC patients have significant dynamics on the wait list. So it could be that things are very stable for a while, but then suddenly there's this spiral, and so a dramatic elevation of the bilirubin over a few month period that can occur. And so I think it's very important for us to understand those dynamics and understand how we can better serve and equitably serve people on the wait list. So this is really my role, and very key collaboration with Dr. Hirschfield here.
I think I was interested in AI and clinical research, and I think PSC, unfortunately, is a disease that's underrepresented, so it felt like a good project to start with.
All right. So I started, like I said, 1990 was my first rotation in my pharmacy program at UCSF. And my first rotation was liver transplant. And the first patient's room I walked into was a PSC patient who got transplanted for cholangiocarcinoma at that time. They were still trying and did not go well. But at that point, it was just one of those things that stuck in my mind of a disease that just felt so different than the rest of liver disease at that point. Continued in my training and then eventually moved over to the industry side and started working. I worked on the oral drugs for hepatitis C. Check. Got a cure there. Worked on new drugs for hepatitis B that didn't cause a resistance. Check. Worked well there. Moved NASH forward. Moved PBC drugs to some extent. Some of them got approved, some of them didn't, but I worked on one that's hopefully about to be approved. And as Gideon said, about 10 years ago, I worked on my first PSC study. And it had always been part of my clinical background, but it also just was so apparent. There was no treatments. It was so tough to enroll. It was, if you looked at where differences could be made, it was really helping clinical trials in PSC look at them differently. Some of the stuff we've learned along the way with Dr. Hirschfield it has changed how we're looking at these future trials, and it's been great to be a part of that. And then I got burnt out on the corporate side and said I want to come back and really get back to the patients, and that's how I landed back here. So that's my story.
Awesome, thank you. Just by the way, if anybody in the room is not into apps and wants to raise their hand for a question, I'll just walk over with the mic. So just want to make sure that we include everybody. All right, so let's do that. Since I said I would, I will do that.
I know that chronic fatigue and brain fog was talked about. And we talk, when you have a general practitioner who in turn talks to the GI doctor, who in turn talks to the hepatologist, how do you make them understand that there is the chronic fatigue and the brain fog, and can you get a prescription for that? Or how do you go about handling all that when you're not directly involved with your hepatologist for your medications?
So, it's difficult. Is the honest answer. Symptoms are silent from a physician's perspective, and we can't see them. And physicians and surgeons are trained to do things for immediate benefit. So they are already primed to know that they don't have the answers that you want. Okay? And then in the context of their day, that makes that consultation actually very difficult for them, because they don't have an answer for you. They don't even understand the mechanism of it. And the mechanism of it is even more complicated because we also know it's not specific to PSC. So there's lots of reasons to be tired or to have a low mood. That's not to say, that's not very important in PSC, but you can see the challenge from the other side of the table is, well, which of these problems should I be responsible as the physician? And so where we've ended up is a trap where people don't want to talk about it, okay? Because fortunately for the doctor, they're so busy with other things that there's enough to fill their day, okay, doing all the paperwork, doing all the emergency letters and prescriptions and sending patients to ER and, giving the antibiotics for the chest infection, that they really are, they can claim quite reasonably they don't have the time. But really what it comes down to is the fact that, we don't have the answers for patients and therefore we avoid talking about it. I think the way to solve that is really, it is about building relationships with your patients and having an opportunity to do the so-called medical and the non-medical aspects. And as a physician, surgeon, whoever you're seeing primary care, acknowledging the importance of the symptom, even if I don't have the answers to the problem. And that's been a strategy that I've tried. It's not always easy. I'm not here to take sides, but you can imagine that if your physician's having a bad day, they're not really in the state of mind to listen to your symptoms. Okay, they won't have that empathy that you need. They're only human as well. So, it may not be every time that you go and talk to your physician, you'll talk about your symptoms, because you might not want to talk about it every time. It may be, you like to bring it up every few consultations, because it's also, it's tiring for you to talk about it, if that makes sense, okay, and not to be heard. So I'm not sure we have the perfect answer, but we are trying.
I think the balance to that is you are your own best advocate. And so if you don't come in, there's lots of resources with tools about symptoms, and the information is great for patients, but especially for physicians who may not be working with PSC on a regular basis, it's great to bring some of that information to them. Some of what we're thinking about is how do you come up with, even if it's just a little list of, here's the most common PSC symptoms, can you just ask about them? Just the simple question of providing that and say, hey, here are things that happen in PSC patients. And if you give a tool, you'd be amazed at how you're making their lives easier to actually check in with you. So it's a joint education around it. But just don't underestimate your voice to make sure that you're actually getting the things addressed that you also need as well. But it is a balance. It's not, the healthcare system is not doing either side any favors right now in terms of being able to spend time that's actually needed with patients, especially around symptoms. It's really tough.
Yeah.
And, you know, to give a plug, we had a fantastic psychology talk, but we heard that they're under-resourced. I mean, every clinic should have a psychologist, whatever the chronic disease is. I mean, and it doesn't have to be a dedicated psychologist. It could be people that the psychologist is then trained in interventions. And you'll hear tomorrow a little bit about one of our intervention studies, which is patient-to-patient peer support. So we're doing a clinical trial run from some European psychology group where we've trained some of our patients to be peer counselors, and our patients are then getting intervention with the peer counselors. It's been a real eye-opener. It's not as easy as it sounds. It really isn't. And, you know, but that data is being amassed. And those are the kind of things that we have to sort of actually generate the evidence for, because then we can go back to the people who pay with the checkbooks and say, well, we've actually shown that it makes a difference, it improves quality of life, and that improves productivity. You can't only fund a new ER. You know, when you build the new ER or when you expand the transplant program with Dr. Selzner, you have to plug in 2 psychologists. Otherwise, you know, it's not a real program. But that's a journey of us advocating.
Thank you, guys. Why was nicotine considered as a potential treatment for PSC? Maybe somebody had a very quick eye and saw that on a screen with all the treatments.
Well, there's a long story there. So, if you're a smoker, you're more likely to have Crohn's disease. If you're a non-smoker, you're more likely to have ulcerative colitis. Okay, and so you know, that observation led people to think about whether nicotine could be used as a therapy for ulcerative colitis. Essentially, it actually does have some impact. It's quite interesting, and it's actually something that speaks to some of the work that Dr. Batt does. And the reason we have to move into this idea of machine learning and artificial intelligence, we really need to capture everything around our patients, not only the things that we think we know are relevant, because there's a lot of information there that we document and tick a box, but we never go and investigate. But if you allow a computer to look at data completely hypothesis-free, it picks up new associations that you didn't even realize. Okay? And they may give you clues. They may not be the answer, but they give you clues to mechanisms. Another one was there was some evidence of the use of certain antidepressants in the fatigue in primary biliary cholangitis, which was picked up from epidemiological data. Now, I don't know where that will go, but it's just the concept that instead of asking questions about 3 blood tests, you know, sodium, creatinine, INR, you know, bilirubin, you know, and expecting that to give you all the answers, what we actually should be doing is saying, well, there's absolutely, you heard it, there's no technological reason why we can't look at all the data now. Genuinely, the technology can look at everything that we hold, you know, and we just have to somehow change how we practice to actually start doing that.
Thank you. We have a question that somebody feels is basic, but maybe it's not. Why is itching and fatigue a symptom of PSC?
Is the speaker mine? Okay, so why is itching and fatigue? So well, we have to divide them into 2, actually. And let's talk about itch first and fatigue separately, because fatigue is just so much more complicated. Itch does mechanistically relate to liver disease, be that hepatitis, inflammation of the liver cells, or damage to the bile ducts. But particularly when you damage the bile ducts, the idea of the bile ducts, they're a complex set of tubes that then lead the bile to leave your liver and go into the bowel and do all sorts of clever things. Okay? But the bile has inherent toxicities because its properties require it to be a detergent in your bowel. What that means is you don't really want bile leaking into your liver and leaking into your circulation. You want your bile going into your bowel, into your stool. When you damage the bile ducts for whatever reason, you get inflammation, autoimmune, ischemia, you know, surgical complications, then you've got leakiness in that system, and the contents of the bile leak out, and something within those contents of the bile leads to irritation. What itch is essentially a form of nerve pain. So the small nerve fibers in your skin are irritated by some substance that's been released from the liver being poorly, in particular something from within the bile. And what happens is that it irritates the small nerve fibers, okay, and it leads to the sensation of itch. And then you've really got the sensation of itch and scratch and itch. And the difference about the itch of cholestasis is that scratching won't help. It will distract you, but it doesn't help the itch. So it's hands and feet, worse at night, feels like ants crawling under your skin. It is, in fact, a neuropathic form of pain. And that's actually one of the reasons one of the sponsors here today, their trial of a drug is actually to target the nerve fiber, not to target the bile acids, whereas other people are working on the bile acids. Fatigue is ever so more complicated, not least because there are lots of reasons to be fatigued. And anyone who's got, PSC, IBD has also got all of those reasons to be fatigued, okay? And they don't go away, and that's called life, okay? But in particular, probably the reason, again, that fatigue is prevalent in our patients is inflammation, and the fact that you need to remember that the brain has been around a lot longer than Toronto General Hospital, okay? So the brain is designed through thousands of years to sense how you're feeling. It's very, very good at picking up what's going on in your body. And one of the responses for the brain at picking up low levels of inflammation will be to feel tired and to not process things. We can't measure that. We have no way of measuring that. The brain has just been doing this for thousands and thousands of years, wherever you are in belief of evolution, okay? Whereas, when you come to the lab and we measure alkaline phosphatase or bile acids, that's really measuring nothing compared to what your body is measuring in terms of your physiology and your function. And so it should, because that's how we've been a successful evolution to get to where we are. So fatigue is much more complicated, but it's also important to recognize that fatigue is not specific to PSC, and there's lots of things you can do about fatigue which are not PSC related. So we've done other studies in a disease called primary biliary cholangitis, and it's true. And you actually, again, I don't want to say how fantastic the psychology talk was, but it was fantastic. We are social animals. And one of the findings that we found, and of course I'm sure the psychologists just knew this, was that the patients who had the greatest fatigue were the patients with the greatest social isolation. Okay? The youngest patients with the greatest social isolation had the highest fatigue burden when they did the scores. We are social creatures. So support groups are a treatment for fatigue. Okay? Contact with people and peers and family are a treatment for fatigue. Again, when I talk about the brain sensing what's going on with inflammation, that's the reason why we encourage people to exercise. I mean, I'm not a leading example of it, but, okay, but, it's true to say exercise is phenomenally good for mild depression and fatigue. Although proving it is actually very difficult, but it's logically, we know that it's true. There are many people from presidents down who use their 5:30 in the morning exercise in the gym to get them through the day and to help, their things. So that's my high-level summary.
I'll just throw one thing, because something that came out of the 800-patient PFDD research was a paper that's going to come out, and they took all the symptoms and kind of mapped them against each other, called a network analysis, and the relationship, because a lot of the buzz has been, well, you have fatigue because you're itching all night, and you have insomnia, and you can't sleep, and it just is this cycle, or it's your IBD, and you've got those symptoms. But what was fascinating in this is it was clear the whole issue of brain fog and fatigue was not strongly linked to either insomnia or pruritus or IBD. So there was a component that is just clear, pure brain fog, fatigue. Both of those were very separate from the others. They were very distinct. And I think that's an important piece to understand. They're probably much more related to inflammation and other things than actually sort of a constellation of symptoms. And I think it's important to recognize it as something very independent. And it may be something either non-interventional, non-drug, or potentially maybe someday a drug that could help with it. But I think, as Gideon said, as a pharmacologist, I will say non-pharmacologic therapy, in my mind, is always a good first step and maybe the best step in many of these things.
Okay, we are over time, but we're going to do one more question because this is fascinating. Can you speak to how your machine learning model might balance some inequalities even among PSC patients, and then it says male, female creatinine, and social economic issues?
Yeah, so that's an excellent question. I think, as Dr. Hirschfield was saying, You know, the beauty of machine learning tools is this ability to take into account multiple variables, hundreds of variables, and longitudinal changes in those variables over time. I think ideally we can provide personalized predictions for an individual patient on the wait list. And with those personalized trajectories, we can also understand, say, what are the top 10 to 20 predictors of that trajectory. So for example, if we anticipate that someone will be relatively stable on the wait list versus someone else may have, say, a certain risk of deterioration, what are those top 10 modifiable and non-modifiable predictors that both our patient as well as the physician can work on? And so that empowers, it provides personalized predictions as well as, you know, points on which the physician can act to improve and change that trajectory, but also empowers our patients. So say if a PSC patient is on the wait list, there is that, say, potential to understand what they might also do as they're awaiting transplant to improve their trajectory. So I think that's the beauty of machine learning tools, is that you're not only taking into account, say, 4 variables to predict trajectories, you're taking into account multiple variables that have changes over time. And the reality is that, so I was just at our Tamardy Center for AI Research and Medicine Symposium 2 days ago, and you know, we were discussing how it's not possible for the human brain to take into account more than, say, 4 to 7 variables at a given time. So you can understand how it's not possible for, you know, a physician to, say, take into account historical changes, dynamics over time in multiple variables. And a computer can actually, so an AI algorithm can actually serve as a co-pilot and help improve physician care of patients. And it's not that the AI algorithm would replace the physician, but rather would help enhance the care that a physician can provide to a patient, and also, you know, PSC or other patients can also feel empowered to understand, you know, what it is they could themselves change about their care so that things can be improved. So I think that's the beauty of, say, applying machine learning algorithms to data that is as complex as what all of us deal with on a day-to-day basis. So the reality is that, say, a physician will see the given patient in front of them, will try to, and will kind of subconsciously integrate a bunch of data, they will try, but a computer can actually take that a step further and, you know, really guide the physician to improve the care that they can provide to their patients. I hope that answers the question.
Thank you so much. And thank you to all of our speakers.